in mathematics, 2002, University of Virginia. Then a master's in the University of California in 2005. And then she has concluded in 2008 a PhD also in the University of California, Santa Barbara. And today we're going to have this talk about the origin of rigidity in biological tissues. And thank you very much, Lisa.
Thanks so much for the introduction. Can you hear me okay? Yeah, thumbs up, it's okay? Okay, before I get started, I just wanted to say how disappointed I am I can't be there in person. I had a personal emergency, which thankfully is on its way to being resolved now. I plan to be visiting Brazil for those of you who are local in May. And those of you who are not local, I hope to see you at another conference. I know how crummy it is when a speaker can't be there in person. I would have loved to have been there in person too, so thanks. And the other thing I want to say before I get started is I want to emphasize sort of the people who did the work I'm about to talk about. In particular, most of the work I'm going to talk about today was done by fantastic postdocs, Ojan Damavandi and Varda Haig. Varda has a job is just about to start at Urbana-Champaign, and so you should definitely check out their work, their career. Tyler Kane is a graduate student in my group who's been working jointly with Chris Santangelo and I on this work as well. Okay, so to start out with, there's been a series of recent biology experiments that say that cells in biological tissues are tuning their properties to alter the global rigidity of the tissue. So let me give you a couple of those examples. This is a paper that's a little older now, like 5 years old now, that demonstrates that there's actually a gradient in tissue fluidity or rigidity across the tail bud of a zebrafish. So up here is the top of the zebrafish. This points towards the head. Down here at the bottom is the tail. And there's actually a more fluid-like nature of the tissue down here at the bottom, and it becomes more solid-like as you go to the top. And Oche and collaborators demonstrated that this is actually really important for body axis elongation in zebrafish. And sort of a similar result is in cultured MDCK cells. So this is some work in collaboration with John Devaney, who's an outstanding graduate student in Margaret Gardell's group. And what we're looking at is the cells just in a petri dish over time. And what you see is that at the beginning of the experiment, let me start at the beginning. At the beginning, the cells are just barely confluent, and they're moving around and changing neighbors every once in a while. And then by the end of the experiment, although cells are still dividing, they're not really moving around and changing neighbors. And so they sort of have a much more solid-like behavior to the tissue. Okay. And so, the other point, I guess, is like, why? Why does biology do these things sort of regularly? And I think the picture I want to leave you with, so here's just a beautiful image of cell division in zebrafish. So this big thing down here is the yolk, and these are the zebrafish cells that are sitting on top of the yolk, and they're dividing and dividing and dividing. But as with many vertebrates, at a certain point right here, the cells actually all start to move and flow over the yolk. And that elongation of the body axis happens, fairly quickly in terms of development. And if you zoom in and you look at the individual nuclei, so these red dots are the nuclei during the stage when this body is moving down across the zebrafish, you see that there's a fair number of cell rearrangements there too. And the idea in general, I think, it's pretty obvious that, as a physicist, that these types of fluid-solid transitions should happen and should be controlled. Because, of course, if you start out spherically symmetric and then you need to have a long body axis, it's going to be really hard to have a long body axis to deform an object really far if it's solid-like, right? So you really need it to turn into a fluid in order to have this really large-scale deformation that's needed for most animals. And then to do things as an animal, right? Like we all do things like, play football or like hold a baby. And those things require you to be solid-like, again, at the end of the day. So we clearly probably need to have these transitions. Okay. And I would say I talked mostly so far about cellularized tissues where all the cells are touching one another and closely packed, but also your body is composed of lots of extracellular matrix, which often exists in disordered networks that also transition from floppy to stiff. So this is some really nice work out of Fred McIntosh's and Heisa Kondering's groups, where you're looking at, so this is an image of a typical, well, reconstituted collagen network. And on this axis is sort of how connected the network is. And what's interesting is you find even in collagen networks that have a very low coordination number, so sort of their branch points, there's only like 3 fibers that meet at a point. So you're over here, and if you just, we'll talk about it in a second, you might expect the system to be floppy over there. It turns out that if you strain the system a lot, so if you take a chunk of this collagen network and strain it a lot, there's a critical point at which all of a sudden its rigidity increases by orders of magnitude. So there's also this floppy to stiff transition in fiber networks that we'll talk about a little later. Okay. And then I don't maybe need to say it for this audience, but I always like this quote by Phil Anderson, which says, of course, we sometimes take emergent mechanics for granted. Like the fact that something is rigid often doesn't surprise us when it actually should. And moreover, it's really kind of an emergent and interesting feature of interactions, of many body interactions between particles or cells or fibers. Okay. So I'm going to give you the canonical example. And again, I know most of you in the audience have probably seen this already, but just so that we're all on the same page, you know, jamming is the canonical sort of thing that becomes rigid at 0 temperature in a disordered way. Okay. And that also, you know, jamming is a good example of our intuition that materials solidify as they become more dense. As either the packing fraction or the number density increases as things become crowded. Okay, and why is that? Well, if I consider the simplest case of frictionless disordered spheres at 0 temperature, then I have the number of particles times the number of dimensions, degrees of freedom, right? That's the number of the ways the particles can move, okay? And when I have 2 particles, when I increase the density and 2 particles start to touch one another, that interaction between the 2 particles introduces a constraint. Okay, so if each particle has little z number of contacts or neighbors that are contacting it, then the total number of constraints in the system, well, is the number of particles, NP, times z, which is this number of contacts per particle. And then I have to divide by 2 because each contact is shared between 2 particles. So then I just set these 2 equal. Basically, I say, okay, we expect rigidity to occur when these 2 quantities are equal. The number of degrees of freedom equals the number of constraints. And then in one line of algebra, you get that this means that happens when the coordination number or the contact number is 2 times the number of dimensions. So if I'm at lower densities, then my number of contacts is less than the 2D and the system is under constrained. And at high densities, my number of contacts is greater than that and the system is over constrained. Okay. And so the other thing I should say is, like, you know, this is a mean field calculation, but it like works surprisingly well for jammed systems. This is exactly what happens, even though it's a mean field understanding. Okay, so I'm going to show it to you in a different way, because actually I'm going to show you how this breaks this down in a minute. So this is going to seem really pedantic, but let me just go through it quickly. So now I'm going to consider the same question for a 2D frame. So this is I'm considering a frame in 2 dimensions, which has sort of these nodes, and in 2 dimensions I have 4 nodes in 2 dimensions, so that's 8 degrees of freedom in the system. And then these bars between these nodes introduce 4 constraints because there's 4 bars. Okay, and then the other thing I should say is, well, there's some trivial motions of the system which are rigid body rotation. So I could have translation in one direction, translation in the other direction, and a rotation. So there's 3 rigid body motions that are allowed that are 0 modes, but I don't care about them. Okay, and so that suggests that there should be 1 non-trivial floppy mode and the system should be under constrained. And I've sort of drawn it in here, which you're all probably familiar with. It's just this box can be sheared, right? Okay, so then if I wanted to make a box that couldn't be sheared, if I wanted to make a box that was rigid and had no non-trivial floppy modes, then I should add a bar, okay? And then I add 1 more constraint, so now I have 5 constraints, and therefore I should have no more floppy modes, and yes, that works. This works well. This is how we engineer things, right? Okay, so there's a little bit of a small extension, but it's pretty obvious, which is that now if I consider 2 boxes, so here's 2 of those same boxes, if I would have been stupid about where I put that extra bar. So here I've been in this picture, which is rigid, I've been smart about where I put that extra bar, right? I put 1 inside each box. but I could have been stupid about where I put that bar. And so if I put both bars in the same box, right, then I would still have a floppy bit on the end, which I hope you can see here. Okay, and so in order to account for that type of behavior, what you can say is actually, if I have a set of tensions or forces on the system that allow all of the nodes to remain in the same place, then that is what's called a state of self-stress. And when I have a state of self-stress like that, I can actually add it into my constraint counting, because it turns out that you can show under some set of assumptions we'll talk about in more detail later, when I have a state of self-stress, that's basically a redundant constraint, and therefore I have an extra floppy mode living in my system. Okay, and so this was worked out by Calladine, and then there's this nice, really, I think, very readable description of the problem by Kane and Levinsky and others. Okay, so, what's the consequence of this sort of constraint counting? So jam packings tune themselves to a highly self-organized state at the rigidity transition. So what I mean by that is, here's a picture of a packing of particles at a density just below the critical density at which they become rigid. And so no particles are overlapping here. And so if I just increase the pressure just a tiny bit, so if I change, my boundary conditions a tiny bit, then all of a sudden, each particle has exactly ZC contacts. It has the mean field prediction of the number of contacts. So this is a picture, and if you go in and look, each particle basically has the 4 contacts that it needs to have in 2D, right across the critical point. Okay, but the key part of this talk that I'm giving is that there's a whole class of materials that rigidify even though they are under constrained. Okay, and so, what causes rigidity in these under constrained systems? And I should say, this work that I'm about to talk about builds upon like a very vast literature that I'll talk about a little, and I'm happy to talk about more as well. But let me tell you what the idea is. Okay, so now here's this picture of this silly 2-dimensional frame again. And we said, okay, we should count the number of degrees of freedom and the number of zero modes and the number of states of self-stress. And but that's not the only way that I can make this frame rigid. Okay, so if I imagine taking this frame and sort of smushing it so that all of the springs become collinear, and I can do that in a nice way, basically by sort of taking the sort of endpoints 1 and 4, and sort of stretching them out like this. And so you can see that if I stretch them out like this, actually, I expect that when they all become linear, the system as a whole becomes rigid when the length between 1 and 4 equals the sum of the other 3 lengths. When they're all collinear, the system actually becomes rigid. And that's interesting because I haven't changed the number of constraints or the degrees of freedom. And somehow the state of self-stress, and you can see that it's a state of self-stress. There's actually, I've drawn in little arrows. There's a set of tensions on all of these bonds, basically, that don't change the locations of the nodes exactly when the system becomes linear. So I have this weird situation where constraint counting, as I just stated before, doesn't work. So let's figure out why it didn't work. Okay, because I thought it should work. Okay, and so here again, I have 8 degrees of freedom because I haven't changed anything. This is still that 2-dimensional frame. Okay, and I have 4 constraints just like I did before. Okay, and I said, oh, I have 1 state of self-stress. Now I can draw this state of self-stress in, and I still have 3 rigid body motions. Okay, and so that tells me I should have 2 non-trivial linear zero modes. If I look at this whole system of equations and include this state of self-stress, I should have 2 zero modes. And in fact, I do have 2 zero modes, and they're demonstrated by these green arrows. The key thing here is that they're only zero modes to linear order in the length of the springs. In other words, if my spring is now along this hypotenuse of this line, then we all know we could do an expansion. That's one of the things we're good at as physicists, that we can show that the constraints only change, that basically increasing this L prime only changes changing L only increases the constraints at second order, right? To this sort of, to this delta. Okay. And so what does that mean? It means that like there should actually be a whole class of materials that behave this way. So that was like a very specific example. Let me give you a different example, which you're maybe more familiar with day to day. So if you think about a guitar string as maybe some spring-like segments, you're all familiar, and the game we're going to play is like, how far apart do I hold the ends of the guitar string? So the guitar string has like a rest length, right? And I can think of it as the sum of the rest lengths of the internal springs if I want. And so if I hold my two fingers, I'm holding the guitar string. If I hold my two fingers close enough together so that they're shorter, the distance between my fingers is shorter than the rest length of the string, then the string is floppy. Exactly when I hold my fingers the distance apart, that's the rest length of the spring, the string starts to become rigid. And if I continue to pull, and that's what you're doing when you tune a guitar, of course, is that I keep actually increasing the effective distance between the ends of the string. And I basically get something that has an increasing shear modulus, right? Which is why you hear the pitch of the tune of the guitar string. Okay. And so, I talked about it in terms of changing the size of the box, right? The size of the distance between my fingers. But I want to make the point now, because it'll show up later, that equivalently, I could think about keeping the box fixed and tuning the internal rest lengths of each one of these springs, because that's just a change of variables. Those two things are equivalent. So if I have a system where I can tune internal parameters, I can think about changing the rest lengths of the individual objects inside of it, or equivalently, I can go across this transition by taking my box and changing its shape. Okay. All right, so I'm actually going to take a moment, because you might say, oh, well, still a guitar string is, a thing we use every day, but it's not, are there any other cases where this happens? And I'm actually not going to play this video by Itai. You can go Google it online. It's very fun. He talks about mechanical metamaterials, and in his hands you might be able to see a piece of origami, and I'll just show you enough of the movie so you can see what happens to this piece of origami when Itai stretches it. They basically, this was a video they made for when they published in Science quite a few years ago now, and you can see that it basically
the particular paper that we wrote in Science deals with the Mira-Ora fold. This is basically a series of parallelograms that, when folded into a sheet of paper, give that paper some mechanical properties that are determined by the fold patterns that we put in to the paper itself. The nice thing about this strategy for giving materials mechanical properties is that by changing the fold patterns, for example, I can give the paper a different effective stiffness. I can also introduce new novel mechanical properties. For example, if I take a banana and I just squish it in my hand, that banana is going to squirt out the other ends. But this particular pattern, if I expand it in one direction, expands in the other direction. And if I contract it, it contracts in the other direction.
Great.
The particular paper that...
let's go past that. So I guess the point that I need to make about that video I just showed you by Itai is that the Mira-Ora fold is an example of a mechanical metamaterial that Chris Santangelo, along with Brian Chen, showed also rigidifies due to this mechanism of second order rigidity. I won't go into the details for this talk, but the thing for this talk I'll say is that it also seems that a diverse set of biological tissues do the same thing. So I told you at the beginning that fiber networks undergo this huge strain stiffening transition. So this is that same data shown in a different way. So now you're looking at the shear modulus of a collagen network as a function of the applied shear strain, how far you've basically sheared the box now instead of dilated or uniaxial. But what you see is this is on a log scale, and so there's a critical strain at about this dashed line at which the system stiffens by orders of magnitude. And so, we think that this is another example, I'll explain why, of a system that rigidifies due to second order rigidity. And the thing that my group has studied a lot is vertex models for biological tissues. So I'll take maybe the next few minutes to talk about what those are. Some of you may have seen them, but some of you may not have. So the idea is in tissues that are confluent, basically, where there are no gaps and no overlaps between the cells. So they're all touching each other. And this is what you see commonly in what are called epithelial cell types that make things like your skin. If you take a bird's eye view and you look down at the top of that epithelial layer, you'll see what looks like a bunch of polygons, a polygonal tiling with no space in between, basically. And so the idea of the vertex model is basically to write down a very kind of simple, and maybe even the right word is stupid, equation for it that basically says, okay, if I look at a cross-section picture like this, and you can say, well, what might I try to measure in a cross-sectional picture like this? I might be able to measure the cross-sectional area of the cell and the cross-sectional perimeter of the cell, because I'm doing a 2D view of this sheet of cells. And so all the vertex model says is, well, let's assume that cells have a characteristic perimeter, P0, that they're trying to achieve, and that there's a quadratic penalty when the perimeter is different from that target perimeter, P0. And so here's the quadratic. And also, let's say cells have a target area that they want to maintain. And then you also pay a quadratic penalty for changes away from that target area. And okay, that's kind of simple. I mean, it makes sense to a physicist, but it's kind of simple. There are sort of deep biological reasons to justify those terms in the equation, and I won't go through it in this talk. I'd be happy to answer questions about it. We've thought about it a lot. But the end result is that if you sort of assume that this is true, then you can make some quantitative predictions. So first, you non-dimensionalize the equations, and you see that there's basically one important parameter, which is called the target shape index, which is just the target perimeter divided by the square root of the target area. It's just the obvious dimensionless parameter. And so if I have something which is more rounded, this number will be small. And if I have something which is like more sort of elongated in general, that number will be a lot larger. So as I go from more rounded to more elongated, that's usually associated with an increase in the shape parameter. Okay, and although it's a very silly model, it predicts basically that there should be a transition of the rigidity of the tissue at a critical value of this shape parameter. Okay, and so I'm going to focus in this talk on the work that was done in the presence of finite fluctuations, because what you see is if you have a model with finite fluctuations, so you just add some noise terms to that energy and forces or gradients of that energy, then you can ask how often in a model tissue using that energy functional, how often do cells change neighbors or rearrange? And what you see is that if the cell shape is below a characteristic value, then the cells don't change neighbors and the system behaves like a solid. And if you're above that critical value, the cells change neighbors and you have behavior like a fluid. Okay, and so again, for this talk, I'm not going to go through the details, but I just want to highlight that you can, this type of model, even though it's kind of extremely simple, makes quantitative predictions that actually match what is seen in developing embryos. So this is some work with Karen Keisha's group at Columbia, a fantastic graduate student, Shun Wong. And this black line here is an a priori theoretical prediction with no fit parameters from these vertex models of where the transition should be between a solid and a fluid. And so in isotropic tissues, there's a characteristic value at which that happens. Again, it's about it's just above 3.8 basically. And so isotropic tissues are basically where the cells aren't aligned at all. They're sort of randomly oriented. We extended the theory in this paper and a few other works in order to see what happens when systems are anisotropic. So either there's anisotropic tensions acting, or a cell is subject, a group of cells is subject to like large shears, because that also tends to align the cells and make the system anisotropic. So in addition to looking at the shape index, the theory also predicts that you have to look at a shape alignment. And so this shape alignment is basically a nematic order parameter, but there's like one little tweak where you have to make sure that you also account for how elongated the cells are. So it's like the nematic order parameter multiplied by like how long the rod is, what the aspect ratio of the rod is for different cells. Okay. But then once you do, and the theory predicts actually once you measure both of these, then actually that there's a curve in space which separates fluid-like behavior from solid-like behavior. And then the data on top of this is from fruit flies, where our collaborator has gone in and actually measured both these sort of specific cell quantifiers that we told them to measure, and also the rates at which cells rearrange. And so that's what this color map on this side is. If the cells aren't rearranging at all, then this is blue. Basically, they're not rearranging. So that's when the tissue is behaving like a solid. And then these warmer colors are where it's behaving like a fluid. And what I hope you see is that this, again, this line has 0 fit parameters. It's not like we predicted this like after we saw the experimental data. This prediction was actually published basically before this paper came out. Most of it was in various other papers. And so this is a priori prediction. And then you can see that it does a really good job of separating the data. So that was a long-winded way of saying vertex models actually are a good model for biological tissues. So we should try to understand why they become rigid too. Okay. So we need a generic way to describe this type of rigidity. And of course, you know, I told you it can't be standard constraint counting that has been used in jamming. Okay, we're going to focus on 0 temperature again because fluctuations are hard. And of course, perhaps you and we too are like, oh, the thing we should look at is basically this like very nice but older work on tensegrity. And there's actually a lot of work, especially by Bob Connolly, has really pioneered thinking about this. And basically the framework that is used by this community is the framework called structural rigidity. And so you basically focus on the geometry of a structure. And so something is called structurally rigid if there's no non-trivial global motion that preserves the constraints. That makes sense, right? Like if you have a bunch of bars and, you know, and strings, if you don't preserve the constraints imposed by those objects, then you expect that you are, you know, if you have to preserve those in the motion, then an object is rigid. Okay. The challenge is that structural rigidity, it turns out, is an NP hard problem, even for simple planar graphs. And I also want to point out to the students in the audience that this was a master's thesis, which is amazing and cool. Okay, so the question is can we come up with simpler tests for rigidity? Okay, and so the obvious one we might think of is what I'll call first order rigidity, which is where there's no non-trivial global motion that preserves the constraints to first order. Of course, if you can do it to first order, you could also do it to second order. So that's called second order rigidity. Okay, and so I'm not going to get into the details because like we're interested in something called energetic rigidity. I'll talk about it in a second. Also, if people want to talk to me about it afterwards, I'm happy to sort of email with people or even Zoom with people. I'm not going to talk about it in this talk, but this might be interesting to experts. Energetic rigidity that I'm about to talk about is not equivalent to structural rigidity. And you can talk to me about why. Okay, I'm not going to go into the details here. So this is maybe a summary, a Venn diagram summary of like what kinds of rigid are possible. And the thing I'm going to focus on today is this biggest category, energetically rigid. And I'm going to ask, when does, for example, second order rigidity imply energetic rigidity? Because it's way easier to check second order rigidity or first order rigidity. Okay, so what is energetic rigidity? It's the thing that physicists do. Okay, so all the things you do is what I'm about to say. So I'm just going to say it. Okay, let's say I have an energy functional that is quadratic and a set of constraints. Okay, so for the obvious thing is like a constraint could be a set of springs. In this case, this F, which is a constraint, would just be the actual length of the spring minus its rest length. And then I square it and I take the sum of all the springs, and that's the energy of a spring network. But it turns out that vertex models have the same form. There's a way of writing origami mechanics that's very similar to this, etc. Okay, so it's a pretty generic form. Okay, we as physicists assume that the system is initialized at a minimum of the energy, which is not something the mathematicians do, which is kind of interesting. Okay, but that means we're sort of assuming our system is mechanically stable at the start. So it's at the bottom of a complex potential energy landscape. Okay, so then of course the obvious thing to do as a physicist is to think about perturbations around the minimum of this energy. Okay, so you perturb the coordinates of the system, and we're going to keep variation in energy to second order. And so when you do that, well, you get sort of a gradient term, and that goes to 0 because you're at this minimum to start. And then we have these other terms, and these other terms are related to the Hessian. Actually, I mean, they are the Hessian, basically. Okay, and so let me go back one slide. The condition for floppiness is just basically a system is floppy if it doesn't cost energy to move it. That's what energetic rigidity is. And it's kind of interesting that it's not exactly equivalent to structural rigidity, because this is the thing I think physicists want to think about. In other words, we're interested in cases where perhaps it doesn't cost any energy to move something, but maybe individual constraints aren't satisfied, but they're interacting in such a way that the total energy doesn't change. That could be possible. Okay. So then we can decompose this Hessian. So you've all seen the Hessian. Maybe you've even seen it written this way, where the Hessian can be written as basically a gram term, which is basically the product of what's called a rigidity matrix with its transpose, plus a pre-stress matrix. So those of you who worked on granular systems or jamming maybe have seen this before in that system too. Okay, this R is, just to be explicit, it's called sometimes the compatibility matrix, sometimes the rigidity matrix. It's basically just the set of first derivatives of the constraints with respect to the degrees of freedom. It's sort of the obvious object in the problem. So what I want to tell you next is that, okay, first order rigidity is exactly the constraint counting that we introduced for jam systems. So let me walk you through that. A first order zero mode is a set of displacements that preserve constraints to the first order. Therefore, you can think of it as being in the right null space of the rigidity matrix. So again, this rigidity matrix is just the derivative of the constraints with respect to the degrees of freedom, right? So if I have a set of displacements that preserve those constraints, it's in the right null space of that matrix. Okay, so that means that if I have a first order zero mode, it satisfies this equation, which is just saying it's in the right null space. Okay. Okay, if there are no first order zero modes, then the system's first order rigid. A state of self-stress is, I told you already, a state of stresses on the bonds of the network that do not result in any displacements. Okay, what that means is basically it's in the left null space of R, right? It's basically, it doesn't change the positions of the nodes. So it has to be in the left null space of R. Okay, and so then you can use the rank nullity theorem and, like, again, one line of algebra to derive this Maxwell-Caledine constraint counting. It is just first order rigidity. Those two are the same thing. Okay, so then second order rigidity is what happens if you do all of this to second order. And in a talk like this, I'm not going to go through all the details, but this paper is published, so you can go have a look. And again, I'm happy to chat about it. The idea is now it's a more complicated set of equations for the second order zero modes. And so if there are no second order zero modes, then we call the system second order rigid. Okay, and so the question which I introduced before, now that we have all that terminology down, the question again is, when does second order rigidity or first order rigidity, which are much easier to check, we have equations for them, when do they imply these NP difficult problems of structural rigidity or energetic rigidity, which is what we actually care about? Okay, when can we get away with a simpler definition? And again, in a talk like this, I'm not going to go through all the math, but basically we've shown in this set of papers that you really just have to look at the behavior of the pre-stress matrix and whether it's positive, definite, or not. At least as a first pass. There's sort of more complicated things that we're thinking about. But basically, if the pre-stress matrix is positive, definite, then we can show that either first order rigidity implies energetic rigidity, or second order rigidity implies energetic rigidity. And I guess there's also like one technical detail that occurs right at the critical point, but it doesn't change this general picture. Okay, if P is this pre-stress matrix is not positive definite, if it has negative eigenvalues, then it's system dependent. So it turns out that there are sort of some examples where you do the numerical calculation, and sometimes the Hessian does have zero eigenmodes and sometimes it doesn't. And we don't have a general theory yet for this case, or when we can place bounds on it yet. The good news is that it turns out that this is enough. So spring networks fall into this class, vertex models fall into this class, and that either either the analytic, or we can show numerically because we have a way to calculate it. That the bottom line is that for vertex models and fiber network models, second order rigidity is sufficient to tell you about energetic rigidity. In other words, fiber network models and vertex models become rigid due to second order rigidity, which is a great thing to know because I wasn't sure why vertex models become rigid. It's because of this. It's because of this sort of second order perturbations to the constraints. Okay, that's basically the punchline of this set of papers. Okay, so what is a consequence? The system is internally tunable. So these materials can tune their rigidity very carefully by making small changes to a continuous internal parameter. So unlike like jammed packings, where you have to change your number of contacts and you get lots of rearrangements and stuff on your way to this final state, in these systems, you keep a totally fixed network topology, always under constraint. And by tuning an internal parameter, you can drive the system very carefully to a rigidity transition. So here's a video of where, let me tell you what I'm doing. I just will pause here. So in this sort of simulation, you're basically just changing the internal rest lengths of all of the springs. And you're basically just changing it until you're basically making sure they get smaller and smaller, the internal rest lengths of the springs, until all of a sudden you hit a point where the system becomes rigid. So here it's floppy, but okay, right there, I think you maybe saw it. So this color is the amount of tension in these bonds. There's a critical point at which the system, right about here, starts to become rigid. And basically it's at a critical value of this internal tuning parameter, which is the rest length of the springs compared to the size of the box. Okay. The other cool thing, and I don't know what to do with this, is that at this transition point, there exists an extensive number of modes that are zero energy to first order, and they all incur an energetic penalty to second order. So the self-organization here is also incredible, right? Like there's like, let's say there are n springs in the system, there are n zero modes to linear order, and they're all of them are penalized exactly at second order. So this is clearly in the space of states of the system is a set of measure zero, but generically we get there by straining the system or changing an internal parameter. And so in the last 5 minutes that I have, I want to tell you about the ongoing work. And so this is work with Professor Chris Santangelo, who's also at Syracuse, and Tyler Hain, who's like an absolutely fantastic graduate student in my group. And we're interested in basically exploring the space of possible states of these sort of disordered networks from this perspective. And so let me tell you the big picture questions and then like a specific example where you can think about this thing more concretely. So the big picture question is, we'd like to know for like these disordered spring networks, is there some kind of mean field description for the geometry at the critical point? Like, what is the characteristic features of this critical manifold in the space of states of the system? And perhaps more practically important, what is the relevant order parameter? Because of course in jamming, you can look at the coordination number. So on one side, on the floppy side of the transition, the particles have no contacts. And then on the rigid side, they each have exactly 4 contacts. So you kind of know what to look for if you're trying to analyze this transition. No such idea exists for this second order transition point. Well, at least not yet, but we have some ideas. Okay, and the second reason we're interested in this is like, we're also interested in designing metamaterials that have specific properties. So let me walk you through a little bit about how we're thinking about this. So I'm showing you another very simple frame. So this is a little different from that 4 frame I showed you. It's still in 2D. But now I have 3 bars, and then I have 2 points that are sort of fixed translationally, but allowed to rotate. Okay, and so we can think about the mechanics of this system as well. And the point is that if I have this setup as shown in this picture, generically, there are no states of self-stress, and there's one linear mode, which is again shear for the system. Again, you can see it by constraint counting. If I'm in this unique geometry where all of the springs are collinear and these 2 points are sort of centered between the rotating points, then this is exactly the same case as the guitar string, and I have one state of self-stress and 2 linear zero modes, but this system is second order rigid. You sort of do stretch the springs to second order when you try to move these things up and down. Okay, the cool thing is that you can see pretty clearly in the system that there's another, because we fixed the topology but not the lengths, there's another set of lengths that you get some really interesting behavior, which is we call a branch point. So this system where the sort of springs are collinear, but there's a point that's beyond where the 2 rotation points are, there's 1 state of self-stress and 2 linear zero modes, and this system is not rigid. And so that's, you can think of it as actually as a branch point, because in general, there's actually sort of this cool mechanical sort of like buckling behavior that these are characteristic of these types of materials. But what I'm showing you here on the left then is a mathematical analysis, because this was such a simple system. There's basically only 3 parameters, which is the lengths of these bonds and this sort of structure. And I'm showing you a representation of the critical manifold of like that basically separates states that are floppy from states that are not in sort of this 3-dimensional space of the lengths. And there's 2 important points here. One is that the states of self-stress, you can prove also, it's kind of maybe obvious from what I've said earlier, those states of self-stress are normal to these critical manifolds that we're interested in. So that's a useful fact. And that there's both a sort of manifold, the ones that are critical are basically where this matrix of derivatives is 0, and that either says the system is rigid or that it's at one of these branch points. And those 2 are sort of separate and different, distinct regions of the space of the configuration of the system. Okay. So then the new idea I think that Tyler has is that we can parameterize the states of self-stress at this critical point. What I mean by that is, okay, we can say, okay, normally we think about writing things down in configuration space, either in terms of the lengths of the bonds or the coordinates of the bonds. So L is the lengths of the bonds and X is the coordinates of the vertices. Okay, and then you also have in these systems that, for example, have periodic tilings or fixed boundary conditions. You also, for finite size systems, you need to describe the boundary conditions. And then this G is just the incidence matrix, which encodes the graph structure of what's connected to what. Okay. Oh, and here's our, matrix. Here's our sort of constraints that appear in the energy functional. The cool thing that Tyler has shown is that actually you can reparameterize everything. So there's a really simple equation for the states of self-stress. So remember, these sigmas are the states of self-stress. This is a pretty easy to compute equation for self-stresses. All possible states of self-stresses that are on the critical manifold for any values of L or any values of the node coordinates. So it's basically a different representation of the basis, basically of the critical manifold in terms of the states of self-stress. And it turns out you can basically like think about walking around in the states of self-stress, and that's a really useful way of thinking about moving around in these systems and also designing in these systems because of this simple parameterization. So what I'm going to show you in the last minute I have is basically, okay, this allows you to easily find states of self-stress. And, it parameterizes all the possible states of self-stress that live on the critical manifold. So you can have systems that are on the critical manifold, and then you can also optimize a different function on those self-stresses. And there's also, by the way, 1-to-1, you can map those states of self-stress back onto the bonds or to the coordinates. So you can get to real space real easy. Okay. And so, for example, one of the ideas is that we can actually optimize the global stiffness of the system as soon as it becomes rigid. So like we're looking for the most rigid, the stiffest critical point on this manifold. Or maybe we want the point on this manifold that can be self-assembled, which means all of the bond lengths are as close as possible to one another. That would be something like this. Or maybe we want an equitension state. And so we now have all of because of this really nice parameterization, we can just do that really easily. And so here are 2 examples of networks, of random networks at the critical point. So this is a picture on the left of a random network that has just been strained to its critical point. So it's one of these like floppy networks that you just strain it until it hits the critical point and becomes rigid. That's on the left. this one on the right is one that we've optimized instead. So it maximizes its stiffness at the critical point. So this is like, it's right at the edge of rigidity, and it's the one that would be most stiff if you pulled it further. You can see it looks quite different in structure to this one. Pretty interestingly, if instead you do the one where you try to optimize so that the springs are all the same length, it really looks a lot like the strained critical network, which is maybe not surprising, maybe it is. And it sort of suggests that maybe there's a way of starting to use this idea that actually this state, which is the equal length state in the state of all possible states of self-stress, is so similar to the strained state that it starts to give you an idea of what might be the relevant order parameters for this transition, which is the one that occurs in biological systems. Should be really interesting. Okay, I'm 2 minutes over. Sorry. So I'll say, okay, in summary, what I'm trying to tell you is that rigidity in many standard materials is described by first order rigidity. So that's like jamming. A large class of mechanical metamaterials are instead rigidified by higher order rigidity. And so they don't require changing of the topology of the network to get to the rigid point. Rigidity in biological tissues, both in confluent cellularized tissues and in extracellular matrix, is described by energetic rigidity, and second order rigidity is a good proxy for energetic rigidity in those cases. In other words, they are second order rigid. The onset of rigidity occurs when a state of self-stress appears due to a very small change in parameters. So for biological tissues, this is a very small change in the target cell shape that can drive you to become rigid. For fiber networks, a very small change to the rest length of the spring can cause you to become rigid, or a change to the strain of the box. Okay, and our current work is focusing on designing multifunctional metamaterials. So they're on the critical point and they are something else, both so we can design cool materials and as a way of thinking about, you know, what we might learn about disordered materials from thinking about the thing that's optimized, that's closest to them. Okay, so with that, I'll leave up my collaborators and take any questions. Thank you so much.
Thank you very much, Lisa, for this wonderful plenary talk. We have time for questions.
Hi, Lisa. It's Carolina Brito. Let me ask you, so you said that vertex model is a very good model for tissues, and you have shown that in a very nice picture. And then you have shown that the vertex models becomes rigid because of the second order rigidity. And if I understand well, you said that as a consequence, you have you can have several floppy modes in the first order meaning of the rigidity. Does it have consequences for the tissues that you are analyzing? And can you imagine an experiment to measure those floppy modes in those tissues?
Yeah, so that's an outstanding question. The short answer is I don't know, because we are still thinking. Well, the first thing is yes, I expect that those zero modes are there, or at least approximately there. Of course, because like perturbations that aren't included in the model might make some of those modes cost a little energy, just like bending modes in fiber networks make that there's a little bit of energy even in the floppy side, but it's small, okay? And I expect that to be true in biological tissues. But the challenge is that it's actually pretty challenging to identify and characterize those zero modes in this disordered networks, because there's an extensive number of them. So there's this subspace of floppiness to first order, and it's hard to sort of decide how to, like, you know, like what basis to use to describe that subspace. So one idea actually that Tyler has had that we haven't had time to explore too much is he has this really cool way of basically a basis of loops inside the tissue. So, you know, each one of these tissues obviously a cell is a loop in the network structure. And it actually turns out that the math becomes really nice if you just write everything in terms of recurrent loops. And so we think that maybe that means that there's actually a zero mode that's associated with each one of those loops. but that is still speculative. And if it were true, I'd love to understand what it means for the biology. It's a great question, and I guess the real answer is I don't know. I just told you my speculation about it.
Yeah, because I guess that I can imagine some movements in the tissues that are floppy modes, but they are not, they keep being rigid anyhow, right? Even though
Yeah.
I see, okay, yeah.
Yeah, so they're rigid to second order, so finite perturbations, but you're right. The idea is they should be sort of cheap, especially for small motion. And I don't know what that means for the biology. You're right, it's interesting.
I have a question. This is Danilo, Lisa. Does it make sense to think about higher order rigidity, like third order, fourth order, and the energetic rigidity being some sort of limiting case for this?
Yeah, so that's a great question. And I have to be completely honest with you that Bob Connolly has written a paper on this that I don't fully understand. I've read it multiple times and I don't fully understand it. What he says in the paper is that third order rigidity is not well defined, and all higher orders above third order rigidity is not well defined, because if you try to basically define it, like, you try to basically ask if something is third order rigid, it no longer maps onto structural rigidity, which was his sort of lodestar for the problem, right? You basically don't, like, third order rigidity, it's both, it has some inconsistencies when you try to define it for sort of, especially like this known set of sort of weird structure, because when you're doing structural rigidity, there's this known set of structures that is known to be weird. And so you always test your theories on that set of frames. And it's sort of inconsistent in the frames and also inconsistent with structural rigidity. So in this paper, Bob Connolly says third order rigidity is meaningless. But I have to tell you, I don't fully understand that paper. I know that's not very helpful. Sorry, Danilo.
Thank you.
Hi, thank you. It's very nice talk, very good talk. Let me ask you just to know if I'm following you correctly. So, did you say that energetic rigidity implies structural rigidity, or are there two different things?
It implies, yeah. So yeah, let me bring up the little pie, because in the paper we try to. Yes, so it goes one way, but this is not a bi-directional arrow.
And
why,
maybe
just oh, go ahead.
No, yes. If that's the case, then in what sense is energetic rigidity more important biologically? I mean, because in a model, the energy is, something you parameterize, you more or less guess, but I would say structural rigidity is more important because it's what you could maybe measure that, right? Why is more important?
No, you're exactly right. So I'll say two things. So I think that's a really good point. So in the case of the vertex model and the fiber network models, it's actually the case that in those, at least in all the configurations we've looked at numerically so far, second order rigidity and also implies structural rigidity. So it implies both, energetic and structural rigidity. So you get both. Okay. Why do we care about energetic rigidity for this paper? Because we were also interested in mechanical metamaterials. And so we thought that it would be really interesting if you could design a material where it cost zero energy to move it, but there were sort of constraints that were changing during that motion. But I agree with you, for biological tissues, it may be less relevant.
Okay, thank you very much.
Does that answer your question?
Yes, thank you.
Okay, I have a question myself. For the vertex model, if you have polydispersity in the shape index, I imagine that even if you have the same average shape index as in the monodispersed case, there might be situations where you are in a different phase. Instead of, solid phase, you are now in a fluid phase because of some sort of, underlocalization, in the sense that part of the system has some shape index, and this is actually what determines the kind of phase that you are. Have you studied these polydispersed systems?
So that's an outstanding question. So Max Dapeng B, who's at Northeastern, has a beautiful, I highly recommend, it's so beautiful and readable paper saying exactly what you just said. Right? Well, let me even be more specific because he had a lovely way of doing it, and I won't give it justice here. But yes, he has a paper on what polydispersity does to this rigidity transition, and he demonstrates that basically it's you can think about, because vertex models are lovely and individual cells become rigid, you can actually just look at percolation of cells that have a shape index above this critical index. So it's not like, you know, granular systems or sort of other systems where you have to do the really difficult rigidity percolation. Just like regular percolation is enough. And so it turns out that the problem's even simpler than you said with like localization and stuff. You get this beautiful collapse of the data if you just look at percolation of cells that have over this critical, individually have over this critical shape threshold. And then maybe just to say one more thing to highlight, I have a postdoc working at my group, Sajjad Arzash, whose work I didn't highlight here, but his work is joined with Andrea Liu, and we're actually thinking about doing physical learning in systems where there's polydispersity. So not only is there polydispersity, but now you're actually allowed to like swap sort of the fluctuation. So you're allowed to basically introduce spatial fluctuations in that polydispersity. And that can also change the rigidity transition. So even imagine you have a fixed amount of polydispersity, but you're just allowed to learn on where to place that polydispersity. You can do even better, which maybe isn't surprising in the end, but it's kind of cool. And we wonder if biological tissues are actually doing that.
It's amazing, thank you. More questions? If not, let's thank Lisa again. Thank you very much, Lisa.